Bueno, buenos días a todas y todos los que están presentes acá. Para mí es un placer recibir al doctor Tony Bean. El doctor Bean vino la semana pasada en ocasión del Congreso Internacional de Maíz. Vino a la Argentina y bueno, tenía planificado una visita y con Ignacio Giampiti, cuando él me avisó que venía Tony para acá, organizamos una breve visita a nuestra facultad y al IFEBA. Hablé con Carlos, bueno, también hablé con los chicos, con Diego Ferraro, con Magdalena, para tratar de justo insertar un seminario en nuestra ronda de seminarios. Y bueno, y Tony muy gentilmente aceptó venir. Estuvimos reunidos antes de venir acá con el grupo Maíz en la cátedra presentándoles nuestras líneas de trabajo. Y bueno, y ahora vamos a escuchar la presentación de Tony. Brevemente, acá ustedes leyeron en la difusión del seminario. Tony es de Canadá, pero en realidad estudió en Canadá, pero luego terminó trabajando en la Universidad de Purdue y ahora está retirado. Es también agricultor en su zona de origen y siempre ha tenido una muy destacada trayectoria en todo lo que es en la parte de ecofisiología de maíz relacionada con prácticas de manejo. Y bueno, son líneas que han sido muy en común con las que desarrollamos los que estamos trabajando, María, Alfredo Cirilo, Fernando Andrade y yo, en el caso de maíz. Así que siempre hemos tenido una muy buena interacción con Tony. Tony no está acá puesto en esta línea de vida que él nos mandó, pero siempre tuvo la gentileza de recibir a estudiantes de grado nuestros y de Balcarce y de Fernando Andrade. No sé cuántos le mandamos y él los recibió a todos y la verdad que todos vinieron muy contentos después de la estadía. Thank you very much, Tony, for the student that I sent you to work with you. They were very happy and enjoy a lot their stay with you there. Y eso es una cosa que para nosotros fue muy importante, que ellos hayan pasado por el equipo de Tony y vieron las similitudes de trabajo y diferencias, y bueno, y eso siempre se capitaliza. Y entonces, bueno, no voy a hablar más, los voy a dejar con Tony y como siempre, acá abajo dice que los que necesiten asistencia mandan el pedido de certificado, ¿no? De asistencia, y después las preguntas Magdalena las va a coordinar. Tony lo va a dar en inglés, pero creo que su presentación, si es como la de Rosario, está toda traducida al castellano por Nacho Giampiti, así que, bueno. Thank you very much, Tony.
Well, thank you very much, Gustavo, and to everyone today for coming and to learn with me. I don't consider myself so much of an expert, even though I've spent 50 years now studying corn. I consider myself a student of corn. But what makes the study of corn so interesting is that the genetics are changing so quickly. And those of us in physiology are always needing to adapt our physiology research to not only the changing genetics, but the changing management scenarios and the changing environments. And so today's talk is going to explore just a few questions relating to efficiency improvements. Because I talked about short corn at the SEAM meeting where we had this huge screen that was 14 meters tall and 10 meters wide, but today's discussion is a lot more intimate because it's a normal screen and and I can see my audience, and so I'm delighted to be able to spend some time with you and to explore some of the questions that even I haven't answered, but which many of you are hopefully going to be excited about exploring in your future, whether it's as a graduate student or whether it's as a professional in the science of crop improvement. So first of all, I'm just going to acknowledge that modern hybrids are different. We know that. The task of a physiologist is always to identify how, when, and why the differences have occurred, and frankly, is from a farmer perspective, is what does it mean for the G by E by M context that we want to address. Before talking about short corn, I want to just mention one of the, will it bother you if I'm here? That's okay? No problem? Okay. I don't want to be too close to you. I'm afraid that I might just kick you or something. So, anyway, so here we are. This is probably the most comprehensive ERA hybrid study that was ever conducted. I've had the opportunity to do ERA studies with Corteva, used to be Pioneer, as well as with Monsanto, which is now, of course, Bayer Crop Science. In my previous work with ERA studies, I would sometimes work with, let's say, 7 hybrids, 1 per decade. And the roots of that go all the way back to my own PhD work with Terry Daynard and Thijs Tollenaar, when we studied old versus new, and we just had 1 old hybrid and 1 new hybrid. Well, this last study that I was involved with up to my retirement, in which I'm still working on as a volunteer, you might say, to complete the graduate programs of students and postdocs, is a study that was conducted in collaboration with Iowa State University and Purdue and the Danforth Center, and it involved 40 hybrids in what we call the short season, which is 103-day hybrids in the northern part of the Corn Belt, and it involved another 40 hybrids in the 110-day maturity zone. Hybrids that were released by Bayer from 1983 to 2020. So it's a very current exploration of the genetics. However, it's important to say that these genetics were before the integration of traits. Okay, so these are, in a sense, the purest hybrid that you can imagine before trait integration. And as we know, whenever we integrate traits, sometimes the yields will go down or sometimes up. But it's a little bit complicated. We measured about 200 plant traits in these studies, and that went all the way from soil analysis to early season probing to root analysis, especially at the R2 stage. It involved biomass measurements, leaf area measurements, leaf appearance rate measurements, and it involved things like nitrogen uptake, whole plant nutrient uptake, ear dimensions, and so forth. Obviously, we only have 1 hour here. I'm not going to talk about 200 traits, and that's not going to be possible. Like any good research, it takes a team that's willing to work together. And in this particular is some of the group members at Iowa State University and those that joined from Purdue. And it involved Bayer Crop Science. And so one of the principal Bayer Crop Science people is Slobodan, who's one of the main breeders, as well as Doug. But aside from those 2 people, the rest of the team was by and large the postdocs, graduate students, and professors at our 2 universities. The first paper that came out of this was on leaf number, and it suggested that hybrids that were released gradually experienced in the last 40 years, between 1983 and 2020, produced leaves faster, closed the canopy faster, Same number of total leaves, reached silking a few days earlier, so that grain fill starts earlier. So that was one of the first papers that came out of that. This research on this very large area study has so far produced 17 papers. There are 3 more in review, and then there's another 5 or 10 that we still hope to write. So obviously, I'm going to be busy in retirement for at least another 2 years. Today, we're only going to talk about one of those 17 papers, and that's the genetic gain by nitrogen rate, because my history of crop physiology studies has always been to involve a nitrogen rate factor as well as plant density, and then to study uptake, remobilization, efficiencies, and so forth. So in this particular study, we only focused on the 110 day maturity hybrids. So there was 38 of those hybrids, and we compared them at low N and high N at more than 10 location site years in our research. The paper that came out of that was this year in the European Journal of Agronomy, and the title, in a sense, says it all. Modern-made hybrids have increased grain yields and efficiency tolerance to nitrogen limitation, to nitrogen-limited conditions. Most of the results always are, in these papers, always focused on genetic gain from 1983 to 2020. And here it shows the genetic gain that was achieved under high nitrogen, and that works out to a gain per year of 122 kilograms per hectare per year. The low nitrogen genetic gain was lower at 88 kilograms per hectare. And if you see a double asterisk here, that means that the difference in the slope between these two is significant. So a lower genetic gain in low nitrogen, but it's certainly the case that genetic gain has improved under both low as well as under a high nitrogen environment. In this particular study, low nitrogen environment meant something between 40 and 90 kilograms per hectare of nitrogen. The average nitrogen rate for low was perhaps about 45 to 50 kilos. The high nitrogen, depending on the field site, was somewhere between 210 and 280. So again, the high averages about 250, more than sufficient for the corn response. I wanted to spend a little bit of time just talking about what were some of the things that were observed. First of all, grain nitrogen concentration is going down, both at the high nitrogen rate as well as at the low nitrogen rate. So that's declining. What is interesting to me, when we're concerned about increasing CO2 concentrations in the atmosphere, and we're concerned about the effects of nitrogen losses to the atmosphere as nitrous oxides and so on, is the fact that we are gaining in terms of grain carbon fixed per unit nitrogen. And so that, to me, was an encouraging sign. So in other words, yes, it's true, we're growing higher yields, but we're not necessarily increasing the requirement for nitrogen. And in fact, we're able to fix more carbon per unit nitrogen in modern hybrids. The other factor that's of note is that the grain nitrogen content that we harvest has increased. Again, there's a difference in slope between the high and the low nitrogen level here. The next comment is going to be on what's happening in terms of harvest index in this 40-year time horizon, and what's happening in terms of total biomass. So the total biomass change is shown here. It's about 60 kilograms per hectare per year. No difference between these slopes at high end and low end. And you recognize that this number 60 is quite a bit lower than the 102 that I mentioned earlier. So the total increase in biomass is less than the actual grain biomass increase, which was 102 at the high end. The harvest index has grown from about 52 to 57 at high end, and from about 50% to perhaps 53% or 54% under low nitrogen. And this is significant. When I was the age of the graduate students that are in this room, I believe that the only pathway to increasing maize yield was increasing total biomass production by maturity, because I believed that harvest index was fixed. And that's not true. And harvest index can increase quite a bit and still have good lodging resistance, still have no problems with harvestability, and still be able to produce an ideal yield. The other thing that has changed is the nitrogen harvest index. And the nitrogen harvest index, in other words, grain nitrogen divided by whole plant nitrogen, is now around 65–66% under low nitrogen and around 63% under high nitrogen. So this is a case where the low nitrogen rate shows an advantage in terms of harvest index. And this means that the nitrogen internal efficiency, or in other words, how much total grain are you producing per kilogram of nitrogen taken up, is quite a bit higher, obviously, under low nitrogen than it is under high nitrogen. But what is of interest to me is that for both of these, there was a change of about 20% in overall nitrogen harvest index. So for everyone who's concerned about how much fertilizer nitrogen we're applying in the United States, and and that we're growing higher yields and farmers are applying more nitrogen, and there's a concern that we're contaminating the environment even more. Well, the truth is that we're actually harvesting more nitrogen in the grain now than we did previously. So from a nitrogen balance point of view, it can result in a positive picture, even though there is a recognition that farmer nitrogen rates now, especially in the last 5 years or so, have started to increase again. They plateaued at about, in the US, at about perhaps 180 kilograms per hectare from the early 1980s through to around 2015. Now they're increasing again a little bit, but still the internal efficiency gains in modern hybrids are very exciting to me. Short stature corn. So now I'm going to talk about some of the same things that I presented at SEAM earlier on. Lodging resistance and late season ground applications are some of the benefits of short stature corn. Higher plant population tolerance, higher narrow row tolerance, higher yields, and higher production efficiencies. So I'm going to primarily deal with these last two. Is it true that short stature corn is going to result in higher yields, and will that also result in higher production efficiencies? And the short stature corn is just shown here by having very little internode elongation, especially in the leaves below the ear. This is just a picture of this past summer of a Bayer Crop Science Preceon with a normal elite tall compared to their reduced brachytic mutation hybrid, which did not lodge when we had wind speeds of 110 kilometers per hour. This is an even more significant wind damage that occurred in Iowa in 2020. And this picture is taken out of their 2022, I think, publication, where they talk about the short stature corn standing up well, even though the tall corn in plots all around it was lodged significantly. And the height reduction does make it possible for later season applications of fungicides as well as fertilizer. So obviously there is a lot less damage being done to the short corn than would be done to this tall corn plant. And fungicide application is becoming increasingly important. And it's estimated that this year, for instance, in Iowa, that about 75% of the total corn acreage received a fungicide application at the R1 stage. So it's about leaf protection. It's also about the expectation that the fungicide may increase the grain filling period duration and may increase yield physiologically, even if there is very little in the way of leaf disease pressure. So fungicide application is becoming, I would say, almost a common practice. Although that does always beg the question, of course, about resistance developing because of overuse of that, chemistry. And that's a concern in weed pests, that's a concern in fungus pests as well. The nitrogen application with ground equipment is shown here with basically side-by-side short corn showing a lot less damage from compared to the in-row application of liquid nitrogen. 28% nitrogen is quite common in the US. I wouldn't say that all farmers are doing this, but when we look at how to improve nitrogen efficiency more, one of the pathways to that is not just a rate reduction, but a split application strategy. And if rainfall is not limiting, then we can get the highest efficiency by applying our last nitrogen application after V10. I wouldn't necessarily want to be this late, but it is a strategy to increase the nitrogen uptake out of the total nitrogen that's applied to a particular field. So this is assisted, you might say, by a short corn strategy. It's very important whenever looking at short corn trials to know what is the source of the genetics. And so most of what we see is that resulting from the genetic mutations. And in fact, the first commercial products are essentially just brachytic, like Bayer Crop Science is thinking about introducing even here in Argentina, is this brachytic mutation. There is also gene insertion, GMO you might call that, as well as gene editing using the CRISPR technology. So the first question to ask whenever you see real results from short corn is, what was the breeding approach that was used to create the short corn? And then secondly, you need to determine after that, what was the mechanism for selecting the tall hybrid comparison? Is it a genetic isoline, or is it just simply an elite commercial hybrid of the same maturity for that environment? Thirdly, you need to understand what is the actual plant density or plant densities that were used in the comparison. Fourthly, you need to know what is the nitrogen rate that was used, or maybe multiple rates, and nitrogen timing. All of these 5 factors are important in evaluating any result of a short corn versus a tall corn. And so that, unfortunately, takes a lot of investment from a research point of view, and it requires many many students to realize as well. So the first paper that Corteva published on their short corn happened to involve gene editing using the D8 technology. So gene edited D8 short corn hybrids. The beauty of this particular paper is that it compared it to the isogenic full stature comparisons. It also acknowledged the density factor. Okay, 8, 10, and 12 plants per square meter, and it had the advantage of 17 locations in a 2-year period. What was the difference in plant height, first of all? And the plant height for the short corn, and short corn I'm going to show mostly in blue today. So if you see a blue bar, that means short corn. So it's about 35% or 32 to 34% lower in height. As far as lodging, that was only measured at 10 locations, and the lodging with the short corn was less. Not zero, even at populations of 8 plants per square meter, but certainly significantly less than it was for the isolines, which were the tall comparison. And it's interesting that at 120,000, the lodging results were not different. Even though the overall average of the 3 densities was significantly lower. So what about yields? Well, the yields were shown in that paper this way. And so they basically said that, okay, no significant difference here. Here there's no significant difference, but presenting it this way just sort of seems like, well, maybe there's 300 kilogram gain there for the short corn hybrid. And maybe here, well, it's not different, but there is something in the neighborhood of another 300 kilogram gain. I never like seeing results presented that way. I'd prefer they use 0 on their y-axis. And if you look at that, everything was 13,000, and it really didn't matter. There's no difference here. Let's just call it no difference, rather than extrapolating and trying to pretend that something is a little taller. Okay, what about Bayer Crop Science? They're most recent. So, okay, this is not publicly available yet, so I got this from a collaborator, Frank Doleman, from Bayer Crop Science in the United States. And again, the advantage of this particular study is that it's biotech short corn compared to isogenic tall corn. It's a 4-year study that I believe was again conducted at least 11 locations site years. And what does it tell you in terms of yield? It tells you that the yields were no different. Both of them were 14.3, 14.5 tons. They go on to say that there is a difference in plant population optimum for the short corn compared to the tall corn. And so the short corn here is shown on the left-hand side, the tall corn is shown on the right-hand side. And what do their numbers show for the medium yield environment? For medium yields, the suggestion here is that the agronomically optimum plant density is 7,000 plants per hectare more. And lo and behold, at the higher yielding environment where you're getting close to 15 tons, the agronomic optimum plant density is as much as 15,000 plants per hectare more. I quibble a little bit with that in part because agronomic optimum is never the same thing as economic optimum. And so obviously it depends a lot on the price of the seed that you're actually having to pay. But it is interesting that in these particular isoline comparisons, there was a higher optimum plant density. That's not always the case, and so I'm going to show some results from Purdue where that wasn't necessarily the case. So I'm going to switch now to the Purdue trials. The Purdue trials were something that I started in 2018. And like you've had to do here, you need to be working with commercial companies because they have the genetic resources, and sometimes they also have the funding. And it's been my experience in mentoring graduate students that you need to find funding from whatever source you can find, whether that is commercial source or whether it's a government source. In the end, you know, when we're in an academic institution, we're about pursuing truth, and we're needing to train the best scientists possible. And so in our trials, even the ones I began back in 2018, I accepted Bayer Crop Science funding in order to do some comparison trials. Full stature versus versus short stature. I'm going to start though with trials that it weren't something I did, but were something that were done by Dan Quinn. Dan Quinn is now the corn extension specialist at Purdue University, and his master's student is Eric Oliva. And so I'm going to talk about the last 2 years of his research. So the first trial is 2 short, 2 full stature hybrids. I should mention that these are based on the traditional breeding, not on, and no isolines here. This is like commercial hybrids. The same maturity, seeding rates of 7.9, 9.4, and 10.9, nitrogen rates 180 to 315. And you may think that sounds a little bit high, and it is a little bit high. And so in the next year, they had same hybrids, seeding rates the same, but the nitrogen rates, they went down to something more reasonable, and they started with 0. And if you don't have a 0 nitrogen rate, you can't study nitrogen recovery efficiency and all of those things. So it's always helpful to have a 0 nitrogen rate. This is Eric Oliva. I think he's originally from Zamorano University. Measured all kinds of traits, including ear position and total height. And always, whenever we measure total height, it's to the top leaf collar. And what was the difference? Well, the difference in plant height was 215 for the full stature, and the short stature was 166, so it's 50 centimeters shorter. Ear height was about 97 versus 57, so in other words, an average of about 40 centimeters shorter ear height. What were the yields in this particular trial? And that's shown over here for the first trial where he compared the 2 short versus the 2 tall, and the yields were lower by about something in the neighborhood of 1 ton per hectare lower. In terms of ear height, again, 90 versus about, let's say, 57 centimeters at the ear position. We employed 3 plant densities, 7.9, 9.4, and 10.9. And what was interesting is that there was a significant interaction in the sense that one of the tall hybrids also increased in yield as the plant density went up. And these are pretty impressive yields, over 19 tons per hectare. And as far as the short corn hybrids were concerned, one of the hybrids also increased in yield as the plant density increased. But you notice with the other full stature and the other short stature that there was actually no increase at all in yield. So sometimes it's true that every genotype needs to be validated if there's no such thing as a general recommendation that all short corn hybrids can be grown at higher densities. Well, sometimes that's true, sometimes that's not true, and it's important to be able to know that with certainty across more environments than are shown even in this preliminary research here. Acre is in 2024, the following year, again, higher yields with the tall hybrids at the bottom here compared to the short corn hybrids. Once again, an advantage of about a ton per hectare for the tall hybrids when averaged over the 3 plant densities. Plant stand is a little bit lower here, and so I'm kind of concerned a little bit about a seed quality issue there. I don't know that it would have been explained a 1 ton per hectare difference, but plant population and final stands are always important to measure. Okay, tall corn. At the second location, we had high wind speeds during late grain fill, and one of the tall hybrids here fell down quite badly, and the yields for that tall hybrid were lower. So here's a loss of about 800 kilograms for this tall hybrid, the one that lodged more. But even then, this tall hybrid, sure, it had lodging damage, but it still yielded about a ton or so more than was the case for the average of the short corn hybrid. So once again, like, lodging is not the only trait that you need to be concerned about. You need to be concerned about the whole package, and the whole package includes questions on optimum plant density and nitrogen, which I'm going to get to in a minute here. So this is an example of the tall hybrid, and just showing the reduction in yield with the probably because of lodging damage. But even in the short corn hybrids here, you can kind of see that, yes, with the one hybrid, we've got an increase in yield with the higher plant density, but that's not true for the second short corn hybrid. And I'm going to switch now to row spacing. And the row spacing research results are only going to be shown here for 2 row spacings, 51 centimeter, and I understand in Argentina you often have 52 centimeters. So this kind of works out pretty close to what might be common here. 76 is the row width that is used on probably 90% of the corn plant acreage in the United States. In this trial, we're only looking at 3 short-stature hybrids at 3 plant densities, 8.4, 10.4, and 12.4 plants per meter squared. And in 2024, again, the same 2 row widths, 1 change in the short corn hybrid, going to a longer day hybrid because of a seed availability issue. These are all with a natural mutation again, all 3 of these short corn hybrids. And again, this was done by the corn extension specialist at Purdue University. This shows the 51 centimeter rows at B10 versus the 76 centimeter rows, showing the earlier complete canopy cover and full light interception. This shows the yield responses. In this case, the 51 is on the left-hand side, the yields with 51. The yields with 76 are shown on the right-hand side, and the individual hybrids are shown with different colors. So this is a 106-day hybrid. The blue is a 112-day hybrid, and the green is 111. And what's striking is that in this site year, this site year, and this site year, so that would be 3 of the 4 site years, we are having higher yields in 51 centimeter rows compared to 76 centimeter rows. And that's the reason why I suggested that in a sense, in Argentina, you might have more to gain with short corn hybrids because of the fact that you're narrower in row width commonly. Whereas in the United States, in order to accomplish that change, it involves a dramatic change in planting and harvesting technologies. Short corn hybrid are, I think, have one negative factor, and that is if there is low rainfall, particularly during vegetative development. And this is a site year with 26 millimeters of rain in June compared to a normal of 100. This is for an environment, by the way, where basically we get 960 millimeters of rain annually, and during the months of May, June, and July, we get typically 100 millimeters per month. So if you have that kind of a situation, you also run the risk of having a pretty low ear height. So here's a hybrid here, only 47. And we get very nervous about being able to harvest the ears if the beginning of the ear height before any drooping occurs at maturity is below 50 centimeters. And so we're kind of concerned about ear positioning in the context of short corn hybrids with early season or even mid-season drought. In terms of efficiency, and this is the final topic for today, are they improved with short corn? Well, certainly from a timing point of view, yes, we can apply some nitrogen fertilizers later, more in synchrony with whole plant uptake. Rate, perhaps there's a possibility of a lower optimum rate. And perhaps there are also, though, from a physiology point of view, differences in remobilization from leaves and stem to the ear. And I understand that's also an interest here in future research at the University of Buenos Aires. And fertilizer recovery efficiency. Well, our first foray into that was based on a collaborative study that was actually referred to this morning by Casella. So Casella is, so in other words, the majority of the authors on this were from Bayer Crop Science, but we had some university representatives from Illinois and from Indiana as well, and some graduate students from Argentina originally, like Leo Almeida Pico. Okay, so 10 field trials, 8 short hybrids. 6 of them were the result of natural breeding, 2 biotech. This, I think Juan, you showed this something, parts of this slide. Where's Juan? Maybe he's not here right now. But anyway, he showed parts of this slide as well. So one way of looking at it at the R1 stage is to look at what is different in the short corn hybrids. So this to the right of 0 would be a positive change. To the left would be a negative change. And you notice that what are the advantages of short corn hybrids? They are higher stock nitrogen, higher ear nitrogen content at the R1 stage, and higher ear nitrogen concentration. Sometimes we notice with short corn hybrids is that they begin to form the ear shoot sooner. They don't necessarily flower sooner, but they begin to put resources into the ear shoot slightly earlier. And we do pick up an advantage sometimes even as early as the R1 stage in terms of ear development. In terms of what happens at the R6 stage, at the R6 stage, the most significant thing that has increased is the stock nitrogen concentration, but you also notice that the stock dry matter is quite a bit lower, as you would expect. Grain nitrogen content about the same, ear nitrogen concentration perhaps a little higher. So this just is a way of picturing what went on with those particular trials. And in that paper, we sort of summarized, the differences in terms of short versus tall at flowering versus at maturity. The differences are sometimes quite apparent even in early vegetative stage. At V10, for instance, there was a 13% higher leaf end content in short corn. Had the same number of leaves, but had higher leaf nitrogen. So this then sort of suggested that we needed to explore more what was going on in terms of remobilization and uptake dynamics in short versus tall. So the study that I'm going to talk about now is a paper that was published in Crop Science. And this is one where the short corn was 25% shorter than the tall corn. Once again, this was done with mutation breeding short corn, not with gene-edited short corn. And this is a comparison where we looked at total grain end content. And because we had measured at the R1 stage, we also determined for each of these three different nitrogen rates, 0, 168, and 224 kilograms per hectare. We also were able to determine how much of the effective nitrogen in the grain is originating from the stem, from remobilization, from the leaves, and how much new N was actually taken up. And this is showing the results averaged over the hybrids. In this particular box, we have the differences between the short and the tall, and there were no interactions here. And what's interesting is that the short corn hybrid essentially took up more nitrogen, new nitrogen, as part of its nitrogen pool that was used to establish the higher grain end content in short corn. So the differences were not because it remobilized more from the stem or remobilized more from the leaf. It's because there was effectively a higher contribution from new end taken up after flowering. And this is the nitrogen harvest index is also higher for short corn than it was for tall corn. And here is the nitrogen recovery efficiency. Here I've got to apologize to you. I said that short is blue. Well, here I've obviously, the next time I give this talk, if you want to come, I'll fix this in the next presentation. Okay, and then roots. So roots are fabulous to study. And for most of the 50 years of my program, I've stayed away from roots because they're too difficult to study. However, it's important to study, and there are certainly people who are enthusiastic about roots, and one of those people that's enthusiastic about roots is Raziel Ordonez. He studied his PhD in Lleida in Spain, and basically he observed that tall corn, was showing the long internodes here, had roots that were not as thick or as much volume as was the case with short corn. And so you can see just how short sometimes these internodes are below the ear. So for the past couple years now, last year and this year, we don't have this year's results yet, but we compared tall corn and short corn at two locations, and short corn now again is in blue, and it shows first of all root mass at 4 nitrogen rates, 0, 100, 200, and 300. And what's interesting about this one is that the short corn had about the same root mass whether you had 0 or whether you had 200 kilograms of nitrogen when sampled at the R2 stage. So this is just fascinating to me. There are no differences if you measure everything at 200, right? So there's nothing there. But this difference early on was kind of interesting. And this shows the root length, and the root length was higher in the short corn. However, everything is affected by what soil do you use. In this particular case, it was a sandy loam soil further south in Indiana, and now there's no differences in root biomass or in root length. And so the difficulty of this kind of research is that the location and the year matters so much. And the frustrating part of root work is that you would have to be prepared to probe in more than one location, and preferably 5 or 10 locations, in order to make a significant conclusion, which is why I spent the majority of my career focusing on above ground traits. Okay, root distribution. Are the roots as deep in short corn as they are in tall corn? Well, we've shown depths here probing down to 120 centimeters or 1.2 meters. And basically, you can see that the short is always is present at all of those depths in both locations. And as far as I can see right now, though individually there's no significant increase in biomass at individual depth increments, I can conclude so far that we're just as deep and have at least as much biomass in the short corn. But to make a result like this significant, I would have to have more location years. And so we'll see what happens in the 2025 results. The other key factor whenever you do roots is you can't ignore the above ground. And so if you're going to study roots, you have to know the above ground biomass and hopefully the above ground nutrient uptake at the same time. And so that's shown here for the above ground crop biomass at R2, and it shows these two low nitrogen rates for the short corn having a higher above ground biomass than the tall corn. And it is shown here at the second location where there was no difference in above ground biomass. And the root shoot ratios, and here it shows that there was an advantage for the root to shoots ratio being higher in short corn, but only when we looked at it at the 0 and the 100 nitrogen rates. So it's very nitrogen rate dependent whenever you look at these ratios. And no difference at all at the second location. So again, these are preliminary studies. This is not published yet because we needed to have at least another year. What about commercial talk on roots in short corn? A paper is now in review, and I'm not a reviewer, for Bayer Crop Science, and basically it has 11 site years that they've looked at across the Midwest. They've studied root presence there, and they measured again root volume. That's shown here at depths from 0 to all the way down to 120, let's say, centimeters. And their conclusion in this paper is 39% higher root volume. That's a huge figure. I don't know. I'm not reviewing the paper, but I would be, I don't know. I would find that, wow, is that real? The second question is, you have to put it in context, right? So if you don't have a above ground measurement at the same time, the root information is interesting but meaningless. Okay, so preliminary conclusions. I need to wrap things up. Can short corn deliver higher yields? Well, sure, it's possible, never guaranteed. Actual yield gains were pretty rare in Purdue trials. Yield gains or losses depend on the genetics, the tall hybrids are compared against, and on the rest of the field management, including nitrogen and density. Some promising opportunities for short corn, higher plant densities, but certainly not for all of the short corn hybrids or production environments. Perhaps more prospect at row widths narrower than 76, and maybe if there is something on root traits that are consistent with the short corn hybrids. Does it give a better efficiency to farmers? Well, yes, it's possible in terms of higher nitrogen efficiencies, and perhaps that could be paired with lower nitrogen fertilizer requirements per unit of yield with no loss in grain protein. So this is a key area for me in terms of nitrogen efficiency, and so there is some possibility there that's certainly worth exploring. There's less likelihood of harvest losses associated with lodging, let's say, during grain fill, but the ear heights can sometimes be too short and cause problems. I'm going to say, this is my last slide. I'm here only because Ignacio Ciampiti, a graduate of this institution, and a former PhD student, said no to coming to Rosario and suggested that I come, even though Ignacio has just a superb group of graduate students and that have finished that, number one, speak Spanish, and number two, are also very good crop physiologists. So I'm here because he said no to coming to Argentina for this particular trip. And like always, everything depends on having a huge team to put things together of graduate students, undergraduate students. And over the years, I've had a number of visiting scholars from Argentina. Many of them are from here. Some are from Rosario. And some of them are from other universities in Argentina. But obviously, it's hard to, you can't accomplish anything without a dedicated group of scientists who are passionate about what they do, who are accurate about what they do, and who are willing to face the challenging questions from their professors. So thank you very much for today. By over-relying on fungicides? Is that your point?
No, when you're talking to use fungicides, you're not considering that the crops can relate with mycorrhiza present in the soil, or that you can add, and the fungicides are affecting them.
Sure, definitely, yeah. So it can actually be a negative for soil microbial life, no question. And it's interesting because the same companies that are selling the fungicides, whether it's Syngenta or Bayer or Corteva, are also the same ones that are investing heavily in bio products, you might say. And so, yeah, I don't know where this is going to end up. There are some products that seem to have efficacy, others perhaps not, but it's no question about the fact that anytime you overly rely on any technology, and especially on a pesticide, that it has a risk of resistance, and it also has, like you pointed out, the risk of being a negative having a negative consequence on the rest of the biosphere. So I accept your point.
But you are not considering to include in your experiment treatments without fungicide and considering roots and mycorrhiza in those roots, or do you evaluate at least?
I won't because I'm retired. But should we? Yes. Yes.
You still haven't seen benefits in your experiment in Indiana, right? With the short. I mean,
from a yield point of view, compared to using the brachytic lines of short corn, compared to elite tall hybrids, we've not seen a yield gain. We've seen, but I'm not too worried about that. I am worried about a yield reduction, and we show that too. So so where I would head with short corn is that if we can produce the same yield but improve our efficiencies, then perhaps there's still an opportunity even though there's not a significant yield gain.
Yeah, but my point is the first goal of the shorter corn,
Right.
dealing with the logic.
Sure.
Then you are trying to find difference in the efficiency, nitrogen use efficiency. But the first goal was to deal with the logic.
Right.
How did you deal with the wind in your experiments? Because you have 2 years, 2 or 3 years of experiment there in Indiana.
Mm-hmm.
But how was the wind in those? Because if you don't have any wind or any significant gas in the experiment,
Right.
maybe you are losing some of the benefits of the shorter corn.
Yes, there was only so I started with this in 2018, and there was only one significant wind event, and that was shown for the southern Indiana location called Butlerville in, I believe it was 2024, and there was 25% lodging in the tall hybrid, but it still yielded more than the short hybrid. So that, yes, it depends how severe the lodging is. It also depends on when the lodging occurs, right? And so there is perhaps more penalty associated with high derecho type winds in the pre-flowering period when the brace roots are still developing. So yes, I hear what you're saying. I don't know. Farmers are going to judge. Like farming is all about risk management, right? And so they're going to look at what's happening in their climate on their soils and judge what the risk is of wind versus another, production risk, right? And so, so in some regions, perhaps that's more important. But I'm not God, so I don't know when it's when it's going to be a problem. But yeah.
Yes, Tony, when I listened to you last week, I was thinking about that. If the technology did not have a high gradient, but also employ a higher plant density, it's not very. Farmers, I'm not sure if they're going to adopt it because they are going to increase the money involved in the production related to plant density.
Right.
Also, if they have to change the machinery to reduce row spacing. So, what is your opinion about the adoption of this technology in the United States?
Well, I would say that the adoption rate will be slow in the next 4 to 5 years for sure. I think that the adoption could be faster if there were regulatory approval for some of the gene edited, but that regulatory approval seems to be sometime in the distant future. And then, yes, this row width thing is a is a big challenge. If because of that preliminary research kind of shows, well, you benefit from the narrow rows with the 3 short corn hybrids that were evaluated. And so there's I don't know, I'm not a marketing specialist, but my sense is that it'll be a niche market opportunity, not a dominant market opportunity, and it also is going to be very genetic base specific in terms of actual improvement, right? So it's
I've got a question because you said short corns work pretty good under 200 kilograms per hectare of nitrogen, and they take a good difference there, and we got more efficiency in the uptake, and this will be here in Argentina. We got high prices of nitrogen, so I think that's a great opportunity
sure.
for short corns and each market. And it's a great uptake in V10, you said, right? Like
We've noticed higher leaf end concentrations as early as V6, and the leaf nitrogen content at the R1 stage is at least as good, and the ear nitrogen content is almost always higher in short corn. And I believe that higher ear nitrogen concentration sometimes, or certainly content, helps to drive a situation where you can increase the endosperm cell number to have an opportunity for filling bigger kernels. And so everything is related to not so much what's going on in the leaf. Yes, it is obviously from photosynthesis point of view, but when you're talking about grain formation, nitrogen in the ear during the lag period is extremely important.
But if I understood well, the higher efficiency of short corn is related to a higher nitrogen uptake after flowering.
Well, it does that too.
Yeah.
It does.
And also a higher recovery of fertilizer.
Yes, correct.
Okay.
Yeah.
Okay.
Both of those.
Okay.
Yeah.
Yeah. Thank you for your presentation. I have a question. Consider the seeding rate here in Argentina, we use lower seeding rates. So what is your opinion about the wheat management or the wheat competition? Because I don't know how is the radiation efficiency interception is the same?
In our experiment, yes.
Okay. So I was thinking if the radiation interception is lower, and then you have problems with the competition.
No, it shouldn't be a problem, and certainly not in your narrow row.
Yeah.
It may be a problem by the end of the crop cycle when you harvest, because leaf losses let the radiation reach the ground, and weeds during the harvest maturity of the crops tends to grow in our form.
Sure. Okay.
So they may leave a field with plenty of weeds.
The short corn, is that?
Yes.
No.
Yes. That's because you harvest them very low.
Okay.
Low.
After harvest.
Yeah,
Okay.
that's it.
Probably.
Especially with harvest delays, with high rainfall periods like you're experiencing
We
in the
may
south.
not use them as a late sown crop.
Right.
They
It might
may be in an early sown.
Right. No, I can understand that.
We may try in Argentina also the possibility to increase square patterns of sowing, reducing the distance between rows, because we have a platform, a mechanical platform that allows us to do that.
Okay.
Even to 35 centimeters.
As I showed
Yes.
25 years ago in my thesis.
Yeah, that's it. So if there is some advantage, we may be found in it using narrow road.
Even narrower than
Even
50, narrower than
the
52.
52 that
Yes,
we're
yes,
using at the
yes.
moment.
Okay.
We have another question?
Yes.
Thank you. Moving away from nitrogen, and I don't know if maybe you can explore tolerance to water deficit, to drought conditions.
Yes.
I was thinking that maybe the lower plants, the short-stature plants, have a lower water loss, evapotranspiration.
Yeah. So the
Level, and maybe they have a different water use efficiency. Have you checked that?
yeah, some of the hybrids may have an advantage there in better radiation or better water use efficiency. I haven't studied that at Purdue because we're in a rain-fed area that is that we often deal with too much rather than too little. I can't answer that right now, but because I don't know. But that would be perhaps a benefit of short corn in terms of less overall water transpiration per unit of biomass gain. So I mean, I don't think we know yet.
I mean, all your trials were under irrigation.
No, all rain-fed. They were all rain-fed, but they were under typically abundant rain, except for the one site year that I pointed out with the very short ear height. But yeah, but it's even in that even in that year, the yields were still lower for the short corn. So it's not automatic that we necessarily gain in water use efficiency just because of having a short hybrid. But when you look at rooting depth is the same, and rooting biomass, if any, and root lengths are higher, then you would hope that there would also be an opportunity to have better drought stress tolerance. But I don't.
In the experiment here from Bayer, evapotranspiration is similar in the short and the tall hybrid, it not change, because you can have, for example, in the root density there, it was very higher than the minimum value to reduce water extraction. Yeah.
Right.
Yeah, above the threshold.
With the same topic, I was surprised that the root, the deepest root, reached 120 centimeters.
Right.
Yes, we have even deeper roots
Yeah.
in Argentina crops. Is that because they are short cycle hybrids, the ones you choose? 102, 100,
No, we had the shortest one we had was 106, and the longest one we had was 116.
Yeah.
So but most of them were right around 110, 112
Yeah.
in terms of. Is it possible for roots to get deeper? Well, maybe if we had done the research in Iowa instead of Indiana, because Iowa has even deeper soils.
Well,
Okay.